It's a pleasure to greet you today for what I believe will be a stimulating session of the International Board, this time about building the future of neuroscience. Needless to say, for me as a neuroscientist, this flagship project is close to my heart. I also know that coping and overcoming the diseases and conditions of the brain is important to people around the world. And I know that so many scientists, especially here at the Weizmann, are very committed to the field and are working very hard trying to reveal its secrets, with focus on how this knowledge can benefit humanity. The wonders of the brain have occupied philosophers for centuries and absorbed psychologists for decades. It is really until just a few decades ago that investigation moved from studying the mind to studying the brain. One could say that while the progress in the field has been phenomenal, neuroscience is still only in its adolescence. Why is the brain still the hardest organ to study when it comes to exploration of the human body? Because its sheer quantity of neurons and complexity of neuronal connections is unlike any network in the human body. It's so complex that it takes 20 years to fully develop, a massive proportion of an average lifespan. Because imaging and other technologies to peer into the brain are relatively new, so the research road is still before us. And also because the diversity of subjects within neuroscience is truly vast. I would say that there is no life or medical sciences field with such a tremendous range of subfields and fields that impact the understanding of the brain. And so, in Weizmann style, when we are faced with important challenges, we can't help ourselves but try to solve them, which is why we are launching the Institute for Brain and Neural Sciences. I will talk more later about the new flagship, but first I want to emphasize again the crucial point around the complexity of the brain and the many areas of exploration, which requires an orchestrated collaboration and a diverse and interdisciplinary scientific community. This video really gives a sense of the breadth and strength of neuroscience research.
Neurobiology studies rely on multidisciplinary, cutting-edge methodologies ranging from molecular biology and genetics, in vitro and in vivo electrophysiology, functional neuroimaging and psychophysics, computational modeling and theory. We investigate how a neural circuit function and guide behavior, how we learn and what we remember, sensory perception and processing, navigation, group behavior, cortical organization, neural coding, synaptic and circuit dynamics, dendritic morphology and function, emotions, neuromodulation, regeneration. Studies are performed on model organisms, from worms to mice, from rats to bats, and even humans. Our students and postdocs are coming from many different disciplines and many different countries. Biology, computer science, physics, mathematics, chemistry, medicine, psychology, engineering, and even philosophy. Here at the Department of Neurobiology, what really matters is that we are doing some of the most exciting neuroscience on the planet.
And that's only the research done in the Department of Neurobiology. In fact, many more scientists at the Weizmann conduct research with relevance to the brain and the nerve system. This is because of the sheer complexity of the human brain in all its wonder.
Hey, Alon.
Hey, Metal, what are you doing up there?
I just wanted to make sure that if you're telling people about the complexity of the brain, that you don't forget to tell them the whole story.
Knowing you, Metal, you are probably talking about sex differences. You'd better explain about that whole added layer of complexity.
Thanks, Alon. You're right. My lab and I are interested in sexual dimorphism, and that refers to just about any difference between the two sexes of a given species. So as you can see all around me in these beautiful examples, it's very obvious in nature that there are differences between the sexes, right? The physiological differences are very apparent. But there aren't only morphological or physical differences. There are also more complex differences in physiology and behavior. And of course, behavior stems from the nervous system. And that's exactly what me and my lab are interested in. We're trying to understand or ask questions relating to the nervous system and how the fact that our brain cells are male or female affect the way they communicate with one another and make connections during normal development. But we also try to understand how sex affects our brain during diseases. And when considering the various diseases that affect the brain, I think that the motivation to study or understand the impact of sex on the nervous system is very clear. Many of the major neurodevelopmental and neuropsychiatric disorders show a strong bias between the sexes. The bias is either at the ratio, how many males are affected compared to females, but also the severity of the symptoms of the diseases. And that could be totally different between males and females. And taking that bias into account, it's clear that if we want to go forward to personalized medicine, any such personalized medicine should consider the sex of the individual. And we are really far from that because for most of these diseases, we don't fully understand the underlying causes. So before we dive into results from my lab, let me just define a few basic terms relating to our brains. There are many types of cells in the brain, but the important ones, the ones that make us who we are neurons. And our brain is what it is because these neurons talk to one another. They communicate. They form connections, what we call networks. And they do so by these specialized structures that neurons have that are called synapses. These are connections between the neurons that allow, not physical connections, but they allow for flow of information from the cell that sends the information to the cell that receives the information. Synapses are really tiny structures that are very hard to visualize. Yet what we can do is to visualize them with tools that revolutionize biology and neurobiology, which are fluorescent proteins. These were proteins that discovered in jellyfish, and they have the unique ability to glow when you shine light at them. The examples that you see here glow in green because they all express a protein called GFP, green fluorescent protein. When we shine blue light at this protein, you can see that it shines back in green. And you can take that protein and tag any protein you want inside a cell, and then you can label an entire organism, such as you see here, flies, worms, and fish, or our entire brains, or even these tiny structures that I talked about, the synapses. And so we took that tool and we made it even more sophisticated. We split the glowing molecule GFP into two, and we attached each half to the different sides of the synapses between the side that sends the information and the receiving side. And only when these two are close enough, just a few nanometers apart, we get that glowing signal, the green glowing signal, which we can image using our microscopes. And so we used that tool and we looked at the exact same neurons in the two sexes, males and females, and synapses between the same neurons. And we saw something that we didn't expect. The connection between the same neurons, but in the two sexes, started as being or looking the same, being neutral. And when we analyzed early during development, but later on, around sexual maturation, so later on during development, the connection started to change, and we saw that some synapses are being removed or are disappearing, depending on the sex of the organism. So if you now don't look only on two neurons, but you look at the entire nervous system, billions of neurons, and you think that separate connections can be eliminated depending on the sex, that's a really sophisticated way to form differential connections and ultimately differential behaviors just based on the sex of that cell. And we didn't just found this developmental process, we also found the exact protein or receptor, if you want, that is responsible for the elimination of the synapses. So that protein, when it's eliminated, is responsible for the removal of the synapses. And these letters you're seeing on the screen are part of the code in that protein. The protein is called DCC, and these letters are the code that the cell needs in order to identify that protein and remove it. And as a consequence, removing the synapses. Now you can appreciate that the sequence is very conserved no matter what organism you're looking at, from a very simple worm to flies to mouse and then ultimately humans. And that is pretty amazing because it allows us to study the mechanism in simpler organisms, but then of course taking the relevance to us humans. And if we jump to humans, mutations in this receptor, in this molecule, DCC, are found in many humans with neurological diseases. And in fact, it's the number one gene or the most affected gene out of all the genes that people have found to affect these neurological diseases. And my lab has decided to focus on one of these, on Parkinson's disease, for a few reasons. One of them, that it's easy to study relatively. There are few models. And the second, that it's differentially affecting the two sexes. So I will end by showing you some recent results we got hot from the oven. The neurons you see here are dopaminergic neurons. These are the neurons that are affected in Parkinson's disease. And the upper panel shows healthy neurons labeled with the same protein we talked about, the green fluorescent protein. In the middle panel, you can see one of the models for Parkinson that we're using. And what happens is that these dopaminergic neurons degenerate, just like happens in humans in Parkinson's disease. But when we force express high quantities of the receptor I just talked about, DCC, you can see that now the neurons look really, really healthy. And so that means that DCC offers some sort of neuroprotection, or it has a neuroprotective role to the neurons. And that's extremely fascinating to us. And we want to understand what's the mechanism. How does it protect the neurons? And of course, the second step would be to also understand whether it affects the two sexes differentially. And with that, I would like to end and summarize that I've shown you today that through basic research, we wish to understand how genetic sex influences normal development, but also neurobiological diseases. You know, I couldn't do this on my own. Alon was right in saying that we need our collaborators in order to further our science. Oh, here comes one of my collaborators now. Hi, Ofer.
Hi, Meital. You know, one of the most interesting questions for me in neurobiology research is how the connections between neurons in the brain allow it to function and perform such complex computations, and how changes in these connections can give rise to a range of psychiatric and neurological disorders. Your research sounds like it's a whole new step in the right direction for answering this. But as Meital said, just like the brain can't function if the connections between the neurons are not right, brain research needs strong connections between scientists as well. My research, for example, would not have been the same without my collaborators in the neurobiology department, like Yaniv, Misha, Tali, Meital, Ehud, and Alon. But I also have collaborators outside the department, like those in the Department of Molecular Genetics, like Iran, molecular cell biology, like Ori, and even outside the department, like Dan and Asaf. Their work has been indispensable for the technologies that we've developed and for the discoveries that we've made. As of my collaborators outside of Weizmann at the Hebrew University, the Technion, Stanford, Harvard, and the University of Toronto, among others.
Hey, wait, am I not at the Department of Neurobiology? Okay, but I have a team of 13 scientists involved in research on ALS disease. If that's not brain research, what is? I want to be moved to Ofer's level on the grid.
Hi, Ran. Good to have you here with us. So my lab is focused on trying to understand a part of the brain called the prefrontal cortex. It's that part of the brain behind your forehead, and what's fascinating about it is that it seems to malfunction in many different psychiatric disorders. We know that the prefrontal cortex is charged with many crucial cognitive functions. These are functions that we regard as higher brain functions. And although we know that this region is important for these functions to operate properly, we actually know very little about how these neural circuits in the prefrontal cortex operate. If we zoom into the prefrontal cortex, we'll find billions of neurons in this area, which are connected with each other in a dense and intricate network of synaptic connections. Yes, just like the ones Meital was talking about. The neurons in your brain are constantly active, even when you're not doing anything, and they communicate with each other by sending chemical messages through these synapses. Our goal as neuroscientists is to understand this code or learn what these activity patterns mean and how they control behavior. We need to understand how these activity patterns change in psychiatric disorders, and if we learn how to communicate with the brain using its own language, these activity patterns, we might be able to design better treatments for psychiatric and neurological diseases. These are major challenges that we're facing, and we can only solve them if we collaborate across fields and disciplines to build an understanding of the brain on multiple levels. I'll give you an example. A few years ago, we learned about a genetic mutation that causes autism in humans. Incidentally, Ori Pellis, our dean of biology, has been studying this gene in his lab and produced some mice that contain this mutation, just like the autistic humans. We looked at how these mice behave and found that they show no interest in engaging in social interactions with other mice. We then looked at the activity of the prefrontal cortex of these mice, and we found something very remarkable. If you present a healthy mouse with another mouse that it's never met before, its prefrontal cortex will display a unique activity pattern that is different from the activity that it would show in response to a non-social object. But the neurons in the prefrontal cortex of these autistic mice, which contained the human mutation in their genome, showed very disordered activity patterns, and they also showed no unique representation for the smell of another mouse. This suggested to us that the autistic brain shows high level of noise in its activity, and this might be the reason why autism is often associated with deficits in attention and why social skills are impaired in autism. When we try to understand what causes the increase in neural noise and the deficit in social behavior, which we see in autism, we need to go back to the synapse. This is this connection between neurons, which allows neurons to communicate with each other. Uncovering the synaptic connectivity matrix of the brain is kind of like finding its blueprint. Brain activity is tightly linked with brain connectivity, and understanding how neurons connect with each other is a major challenge in brain research. We know that learning involves changes in these connections, and we also have indications that synaptic connections are impaired in psychiatric disorders, but we don't really understand the nature of this change or what it means for the function of the brain. In order to understand how neurons connect with each other, we need to be able to activate them and record their neighbors. This way we can find who is connected with whom and what kind of messages they transmit. To do this, we need a technique that can allow us to stimulate individual neurons while we monitor the activity of all the others. Essentially, this is a way to target individual neurons in the circuit, tag them, and somehow be able to control their activity. So for this, we've been developing a technology that's called single-cell optogenetics. The idea is pretty simple. You make the neurons sensitive to light, and this can be done by inserting little pieces of genes that cause the neurons to express light-activated receptors that we call channel rhodopsins. Once a neuron expresses this gene, it becomes light-sensitive. And then we need to be able to shine light on it, which will cause it to become active and release its neurotransmitter message to all of its connected partners. If we can listen carefully to what the neurons are doing, we can actually discover in this way which neurons respond to which other ones. And bit by bit, using this method, we can plot the entire structure of the network. So how can we bring a focused beam of light to something as small as a single neuron? Here we are relying on technology that was developed just down the road in Dan Aron's lab at the physics department. Dan developed a way to create very fine holograms using a laser and some simple optical elements. And if we combine this technique with our optogenetically capable neurons, we can tickle them to become active very precisely. And in this way, measure their synaptic connections and start to draw out the structure of this very complex network in a way that's never been possible before. We've already used this technique to study the connections between neurons in the prefrontal cortex that interact with the amygdala. The amygdala, as you probably know, is a hub for emotional processing in the brain, and we've shown that its interactions with the prefrontal cortex are crucial for coping with fear and anxiety. By mapping the connections between prefrontal neurons that interact with the amygdala, we found that they generate a specialized network that can transmit messages very efficiently. Our goal in the future will be to understand what these messages are and how they regulate emotional processes like fear and anxiety. As you probably noticed, I'm really excited about these findings and about the technologies that we've been developing and delivering to the world.
That's really exciting, Ofer. Thanks. The optogenetics tool that Ofer is working on will be part of the Center for the Development of Innovative Neurotechnologies within the new flagship project. The new tools for exploring the brain that will emerge from that center will enable so much of the research that will be done across the entire entity. The project will include 11 centers dedicated to mental and emotional health, neurodegenerative research, brain injury and regeneration, the way the brain perceives and causes us to act and move, and many more. All this activity requires collaboration at the whole new level, so that scientists in the different subfields begin to speak the same language and exchange ideas. I believe you noticed the importance of collaborative, multidisciplinary work in Meital's and Ofer's presentations. To achieve our goal in neuroscience, this cross-breeding of ideas is essential. We need to build the physical infrastructure to enable that new reality and a major funding mechanisms to scale up what we have today. The Institute for Brain and Neural Sciences is a $200 million project for which half of the fund will go to a state-of-the-art building. The building will bring together about 40 research groups across campus, and just like Eran mentioned, it's not just member of the Department of Neurobiology. So I guess we will have to expand that opening video. The building will physically interconnect scientists in its different levels and sections, and in doing so, be a physical manifestation of the interconnections and level of the human brain. It will take several years to construct the building, 6 to be exact, but I'm looking forward excitedly to its future.